Hey everyone, I'm Shuze Wang. I'm a fourth-year PhD student at University of Michigan. Today I'm going to talk about opposing gradient of retinoic acid and sonic hedgehog specified tonotopic identity in the mouse cochlea. Hearing is mediated by a specialized sensory epithelium, the organ of Corti, which is located in the cochlea of the inner ear. The organ of Corti is stretched along the apex to base tonotopic axis of the cochlea. High frequencies are detected in the base, while low frequencies are detected in the apex. Generally, frequency tuning requires a specific set of proteins to be expressed with tonotopic gradients. Therefore, our aim is to identify the molecular mechanism that confers spatial identity during cochlear duct extension. During embryonic development of mice, a group of primordial cells at the base of the cochlear ducts divides, and thereby contributes to the elongation of the organ. Around embryonic day E12.5, apical cells are born first, and during the following 48 hours, the primordial give rise to new cells that continuously elongate the cochlear duct until the final size is reached. To answer the question, we propose two hypotheses of cochlear duct extension. The first model is called time-space translation model. Time-space translation model is strictly controlled by time, which translates development time into spatial information. Specifically, the E12.5 apical cells are similar as the E14.5 apical cells, and the E12.5 basal cells are similar as the E14.5 mid-basal cells. The second model is called dynamic morphogen model, which morphogen-based signaling originating from the opposing end of the cochlear duct could potentially confer spatial information as well. Specifically, the E12.5 apical cells are similar as the E14.5 apical cells, and E12.5 basal cells are similar as the E14.5 basal cells. The major difference between these two models is whether the E12.5 basal cells are more similar as the E14.5 mid-basal cells or basal cells. To test the hypothesis, we generate single-cell RNA-seq data from the embryonic cochlear duct and spatially reconstruct the organ to investigate gene expression gradient. We analyze single-cell RNA-seq data of the developing cochlear duct across two different time points, E12.5 and E14.5. Here we summarize the cell number for each time point. Next, we identified clusters at annotated cell type for each dataset respectively. By concentrating on cochlear duct cells, including cochlear floor and cochlear roof cells, we reconstructed the 2D and 3D cochlear duct. During development, the cochlear duct segregates into spatial domains asymmetrically arranged along the cochlear duct circumsphere. Here we generated a schematic representation of the asymmetric organization of the developing cochlea. For spatial reconstruction, PCA was performed. The results were projected onto 2D scaled PCA plots. From the 2D projection, the x-axis distinguishes the cell types and the y-axis represents the tonotopic axis with apex facing up. The 3D anatomy of the cochlear ducts were reconstructed by projecting the 2D PCA onto a cylinder surface, whereby the head of the cylinder was representing the tonotopic axis of the developing cochlear duct. For better visualization, we generated a circular projection by flattening the apex base axis, which resolves the asymmetric organization of the cochlea. To validate the 2D reconstruction, marker genes were plotted onto the circular plots. To test our hypothesis, we focused on the cochlear floor cells and first identified the gradually expressed genes along the tonotopic axis. Here we show the overlapped gradually expressed genes between E12.5 and E14.5 data using heat maps. Next, We use the gradually expressed genes as feature to test our hypothesis. We aggregated 10 cells as a metacell and calculated the similarity matrix for pairwise metacell comparison. From the heat map, it shows that E12.5 basal cells are aligned with E14.5 basal cells, which supports the dynamic morphogen model. To test the robustness of our finding, various parameters that were used to generate the affinity matrix were adjusted. The results are consistent across various parameters. Overall, the findings strongly support the dynamic morphogen model. Next, what are the morphogens? According to the previous publications, in the mouse cochlea, SHH sonic hedgehog mediates apical identity, and RA retinoic acid signaling patterns the basal identity, and therefore we hypothesize that RA and SHH form an equilibrium of two opposing morphogen gradients, which jointly control the tonotopic patterning of the cochlear duct. To test the hypothesis, first, the differential expression of HMGA2 along the tonotopic axis was confirmed for the DMSO control. HMGA2 is a reporter gene. Next, explants were supplemented with ectopic RA, which resulted significant reduction of HMGA2 expression in the apical compartment. Conversely, cultured with RA receptor antagonists significantly increased HMGA2 transcript level at the base. Activation of SHH increases HMGA2 expression in the basal compartment, which was similar to the observation after blocking RH signal transduction. On the other hand, supplementation with SHH antagonists significantly decreased HMGA2 expression comparable to the level seen after application of the ectopic RA. In summary, we reconstructed 3D developing cochlea from single-cell RNA-seq data. The hypothesis testing predicted that morphogens encode spatial information. Also, mirrored phenotypes suggest an equilibrium of RA and SHH controls tonotopic identity. And here I would love to thank everyone in our lab, especially Saikat, who did all of the wet lab experiments to support our ideas. And thank you so much for listening. I'm happy to take any questions.